As promised, today's review session is going to be pretty intense because we're going to cover the neurobiology and brain anatomy. Nothing new here, everything that we covered in class. So, oh, see, good thing. All right, I'm going to start the recording. Hang on 1 second. Hopefully I won't have to sign up this time. Okay, so thanks to Allie, we are now starting the recording, which thank you so much for reminding me to do that. I always forget. So yeah, let's just get started talking about neurobiology. First thing we're going to talk about, of course, is neurotransmission. Now, I have made the assumption that most of you have a pretty good understanding, because we've repeated so many times through so many different units, of how a neuron transmits its information to another neuron. I think that's the basic stuff. If not, you don't feel like putting it in the chat window, maybe you could send me a private message. I don't know, maybe I should have taken a private poll. Just to see how much detail I need to go into this, because I could spend all day just talking about how a neuron transmits its information. But that, as we know, it came up over and over again in different units. That's something that's really fundamental to the understanding of everything in psychology, because if we understand how the neural impulses get transmitted around our brain, not just our brain, around our whole body, then we know where to interfere with the science of psychology. Excuse me. So, with neural communication, we know that that human brains are different than any other brain in the animal kingdom, and a lot of that has to do with that area in the prefrontal cortex. Most animals have highly evolved motor areas and sensory areas, like a cat. Tons of motor areas, tons of sensory areas. That's why they say cat-like reflexes. And the association areas, where it makes memories and does that longitudinal thinking, just don't exist in a cat, but they do in a human. So this is the neuron. So I'm going to move kind of quickly through this, but by all means, if anybody needs me to slow down or needs a better explanation or a different explanation, just please let me know. So we've got, on the left-hand side here, we've got the dendrites sticking out of the soma, or the cell body, and the dendrites connect to an axon terminal from a transmitting neuron. So they receive a signal through the dendrites, which are the nodules that stick out of the soma on the left-hand side of this image. And they transmit that signal down into the cell body, which interprets that signal and figures out what type of neurotransmitter it needs to release from which axons in its axon terminals, which axon terminals at the end of its axons. So then, when it determines how it needs to fire, it then sends that action potential, or that electrical impulse, down the axon, underneath these things called the myelin sheath, which are basically like the insulation on the outside of an electrical cord. They keep the action potential from losing any of its strength as it goes down. And when the action potential gets down to one of the end of these nodules on the right-hand side, the terminal branches of the axon, it triggers a little, and I use the metaphor of a water balloon inside the axon terminal, triggers one of those to pop. The water balloon, of course, contains neurotransmitters, which then get released out into the synaptic gap, and out into the synaptic gap here. And then the neurotransmitters bind to the receiving sites on the dendrite. Again, when enough of them bind, it causes that next dendrite to tell its soma, or cell body, that it needs to fire an action potential. If not enough of the neurotransmitters bind to the receptor sites on the dendrites, then the action potential stops right there. So this is where psychologists can get involved in especially with drugs like we talked about today in class, because we can influence the transmission of these neurotransmitters. So I think everybody has a pretty good understanding of that. If you don't, again, let me know. Just going back a little bit here. So this is where I started to go really quick. So that is how the action potential gets from the receiving ends of the dendrites all the way down to the terminal branches of the axons. We have different types of neurons, right? We have sensory neurons. Those exist all throughout our body, mostly in our skin cells. They carry messages from the body's tissue and sensory organs inward into the brain. We have motor neurons, which carry the messages from our brains out to our bodies and tell our muscles how to move. And then we have interneurons, which are the brain's internal communication networks. Where do the interneurons exist? Only in the brain. You won't find any interneurons anywhere else besides in the brain. The body has a few million sensory and motor neurons, but it has billions and billions of interneurons, so many that we can't count. We have no idea how many brain cells we have. We just know that we've got billions and billions of them. So the parts, we talked about this, the cell body, that's the life support center. That's, you know, all the basic life cell parts that you learn in biology class. The cell body here is no different than any of the other cells that you might find. Back up, please. So, so that cell body has got all the major parts. You don't have to know any of those, you know, but it produces energy. It consumes glucose. It produces energy, and it thinks, but it doesn't think. You know, it thinks through chromosomal. You back, please. Yeah, I got it. Thank you. Okay. So yeah, so the cell body, and then it's got the dendrites that branch out from the cell body. Those are the receiving sites, and then the axon is that long extension cord that runs out the bottom of the cell body to the axon terminals. The myelin sheath is that coating that exists on the outside of the axon, and that acts as like the insulator for the electric cord, extension cord. And then you've got the terminal branches of the axon. That's where the places where the neurotransmitters are contained and where the neurotransmitters are released from. Just another view of it. Now the action potential, you may get a curveball question on action potential, but I would not waste my time really studying how the action potential moves. In fact, I've got, see what AP cares about. Doesn't say anything about the movement of the action potential. Doesn't say how the action potential originates, and you're probably not going to get any questions on this, the potassium ions, sodium ions. I'll leave this up for a second because I know that there's some people that are screenshotting. If you want this information, great. This isn't even information that I made you have in your notes because it's just kind of an FYI if you're interested in how the signal gets transmitted down the axon. It is kind of like an electrical charge in that there's the movement of ions, but it's not your typical electrical charge like electricity that you learned in chemistry class, where there's an exchange or a movement of electrons. It's just a little bit different in the way that the electrical charge is transmitted. So we're going to move. What do we need to know? I don't even think you're going to have to know about movement of ions. I think all you're going to have to know is what an action potential is, how it gets generated, and the purpose of it. So really, if you know that the dendrite receives the neurotransmitters, tells the cell body to fire, and that firing is actually an action potential, which is an electrical charge that travels down the axon and tells the axon terminals to release neurotransmitters. I think that's basically, if you understand that, you understand how that works, and you understand what parts of the cell anatomy are involved in each one of those processes, I think that's basically all you'll need to know. There may be a curveball question that asks something about the movement of ions in and out of the axon, but I can't remember ever seeing anything like that. And if there was something, it might just be one of the 100 multiple choice. I hope I don't kick myself later, but I'm pretty confident looking at all the released FRQs that I could get my hands on that you're never going to see an FRQ that deals with action potentials. So, wasn't there another one? Oh, just okay. All right. So again, that's just for your FYI. You do have to know that action potentials are all and none responses. So either the axon, either the neuron fires or it doesn't. This all or none response I have seen before. So so maybe something about a question about maybe the intensity of how a neuron fires or something like that. The intensity is maintained the same throughout the entire length of the axon. It doesn't slow down. It's all or none. It doesn't partially fire. It doesn't. It either fires or it doesn't. So pretty easy. So yeah, in class we went over this, and I asked you what the what because it's really kind of it's a complicated thing to go through it, and something that's well beyond what AP is going to ask you. So we just need to look at how this action potential. The majority of the questions, if from this unit at all, are going to be on what happens inside that synaptic gap. That's the most important part. It's what happens with the release of the neurotransmitters and the binding of those neurotransmitters to the receptor sites on the dendrites. If you understand that, then I think you've got the bulk of this unit down. So again, the neurotransmitters travel across the synapse, bind to the receptor sites, and then influencing it to generate its own action potential. So really, you're just getting action potential to action potential to action potential. You're just getting all these neurons firing in various different ways. But of course, when one neuron fires, it connects to thousands of neurons, and then when those neurons fire, those neurons connect to thousands of neurons. So the action potentials get multiplied exponentially very, very quickly throughout the interneurons in your brain. Then we've got reuptake. So we've got the neurotransmitters that are released out into the synaptic gap, and they bind to the dendrite, and they tell the receiving cell to fire. Now, what do we do with all those neurotransmitters that didn't bind to the dendrites? They're just floating around out there in the synaptic gap. If the neuron leaves them out there, then there's a chance that they could potentially make the neuron fire again by mistake. So once these action potentials, or once these neurotransmitters that are sitting in this diagram, you see them sitting inside the receptor site in the dendrite, they eventually get absorbed into that dendrite, and they become part of that dendrite, and then eventually get, I guess, would get excreted by the cell somehow. But they get the cell actually absorbs them, which leaves this receptor site open again. So if you've got these neurotransmitters bouncing around in the synaptic gap, then there's an opportunity that they could potentially make this dendrite fire again when maybe the sending neuron doesn't want it to fire. So maybe it just wants it to fire once. Then what do we do with all these extra neurotransmitters out in the synaptic gap? Well, we have to get those out of the synaptic gap somehow, so we don't have accidental firing. And that's where the reuptake takes place. So it sucks those neurotransmitters that aren't used back up into the axon terminal, makes a new kind of metaphorical water balloon around them, and keeps them in the end of the axon terminal until another action potential tells them to fire again. So this is obviously this number one here, the synthesis and storage of neurotransmitters. Obviously, if the dendrite's going to receive them and not give them back, then there needs to be a constant process of synthesizing neurotransmitters inside the end of the axon terminal. So so that's definitely one of the main goals of the axon terminal, is to synthesize and then release. This is just another look at that, of how the neurotransmitters travel across the synaptic gap. And of course, we talked about, this is something we talked about today. We talked about it in several other units as well. It's the SSRIs of Paxil, Zoloft, and Prozac, and they get inside that synaptic gap, and they don't allow the axon to reabsorb in this case, serotonin. It's just, to me, it's just amazing that we can chemically make drugs that would interfere with just the reuptake process of the axon terminal. It doesn't interfere with the release of the neurons, it just interferes with the reabsorption of those neurotransmitters. I said neurons, neurotransmitters. It's just amazing that we can get drugs that are that chemically specific, that can get in there and act as an SSRI. So come a long way. Now, if you have the ability to screenshot, this is probably one you want to screenshot. Although I think this is in your notes as well. And there's really no way for me to help you get this down other than when I first presented this to you, you know, I told you these are the neurotransmitters that will, you know, if there's going to be questions about neurotransmitters, specific neurotransmitters on the AP test, I can guarantee it's going to come from this list. Well, I can't guarantee. I can never guarantee anything with the AP test, but chances are, if they're going to ask about specific neurotransmitters, it's going to be these specific neurotransmitters. So I have a pretty good confidence level that if you know these 6, acetylcholine, dopamine, norepinephrine, serotonin, GABA, and endorphins, if you know what those are, you know the affected functions, and you know some of the associated problems with them, then I think you'll be able to ace any question that has to do with neurotransmitters, specific neurotransmitters on the test. And, you know, it could be a question like, you know, a multiple choice question that says something like Parkinson's is a disease that affects arousal, it affects motor neural communication, and it can be caused by an excess of what neurotransmitter? And then you would obviously have to get dopamine, or it would be a question like a lack of serotonin can have an effect on sleep and hunger and is often associated with what psychological disorder? And then you would have to pick, you know, depression or anxiety from the list. So asking questions either way, but chances are you're going to get some neurotransmitter questions. And if you do, I think if you know these, you'll be pretty set. How you get these into your memory, that's up to you. I don't know if you want to use a mnemonic or some sort of studying device, but for this unit, this is a chart that you need to spend a little bit of time trying to commit to memory as best as possible. Questions on that? Good. All right. Parkinson's disease is a disease that we covered in class just to show what happens when neurotransmitters kind of go out of whack. And in this, Parkinson's is caused by basically the brain starts pruning neurons that produce the neurotransmitter dopamine. So we get this lack of dopamine. And unfortunately, dopamine is a drug that we don't have a whole lot of agonists for, so we can't go in and chemically reproduce the neurotransmission that would take place with dopamine. So again, stem cell research may get us there eventually, but right now Parkinson's is not a disease that we can cure. We can treat some of the symptoms, but it's not really a disease we can cure. And these are the types of drugs that can be entered into the synaptic app. We've got agonists, they mimic. The antagonists block, and then the reuptakes. So really, when we have a drug that affects the dendrite, the receiving part of the neurotransmission, it's either going to be an agonist or an antagonist. And you see here the blue up in the top. That's the lock and key mechanism for how a neurotransmitter sits in the receptor site of the dendrite. We can chemically reproduce an agonist that mimics that neurotransmitter enough so that it can get the receiving end of the dendrite to think that neurotransmitter is sitting in that receptor site, causing the neuron to fire. Or we can get an agonist that sits in just enough so that no other neurotransmitters can get there, but it's not enough to make the dendrite feel that there's a neurotransmitter in the receptor site. So we can either block neurotransmitters if you've got too much of them, or we can mimic them if you don't have enough, which again is kind of cool that we can get chemicals that are so synthetically superior that we can actually get in there and influence just the receptor sites of the dendrites of the neurons influence neural communication. It's just really cool that we've come that far. So, neurotransmission summary. Dendrites have three parts. Neurons have three parts: the dendrite, the axon, and the soma, or the cell body. They communicate with each other at junctions called synapses. The flow of information across the synapse is unidirectional. The dendrite cannot communicate with the axon. The axon can only communicate with the dendrite. Neurons undergo electrical events called action potentials. Neurotransmitters are released following an action potential, and the effects of the neurotransmitter on the postsynaptic cell are terminated by the uptake process. If you understand these bullet points, and you understand the cellular, the anatomy of the cells that are responsible for the anatomical features of the cells that are responsible for all of these things happening, and you understand how all of this stuff happens and the details about this, then you're pretty good with neural transmission. So I'm going to assume that everybody does. If you don't, or if there's a part that I need to go back and explain, feel free to stop me. And if you don't want the message to appear to everyone, again, you can just, over here on the left-hand side, just click that send to everyone, and you could just send me a private message if you want. If you want me to go back, just so you know, you can do it anonymously if you'd like. I guess it would be anonymously because I would know about it. But assuming everybody's good with neural transmission, we're going to move on to the nervous system. Move really kind of quick through this because there's really just two parts. It's the central nervous system and the peripheral nervous system. The central nervous system on the guy on the left here, gal maybe, who knows, is shaded in red, and it's just the brain and spinal cord. And then the peripheral nervous system are the nerves that are shaded in orange on the right-hand individual. And extension cords, and bundling them all together with big, huge wire ties. So you've got this gigantic cable, but inside that cable, you've got thousands of different cords that can each send their own individual signal. That's what a nerve is, and a nerve is something we can actually physically see with our naked eye, because there's so many axons bundled together. When each axon can only be seen at the microscopic level because they're cells. But when you put enough cells together and you organize them together in such a way that they build up this mass that you can actually see with the naked eye. So the did I say that? Oh, so yeah, a nerve is the axons, right? So it is the body's speedy electrochemical communication system. Very, very fast. These neurons transmit information extremely quick. And then we've got the central, which is the brain and spinal cord, and the peripheral, which is everything else. I gave you this chart because I think it does a really good job explaining what the different parts of the nervous system are all about. Neural networks are just interconnected neurons. So it's when you think of a neural network, it's all of the axons connected to the dendrites, and all of those making all those different thousands of different connections. On the bottom, you've got an actual slide. Actually, I gave you, Cassie, I gave you that chart. That was in the handout packet that I gave you today. But if you want to take a picture, go for it. If you want a digital copy of it, I'll sit here for just a second. Never mind. Okay, good. All right. So down on the bottom is a microscopic slide of neural communication of the neurons and the different connections that they make. It's exponential. When you talk about billions and billions of these neurons connecting, each neuron connecting to thousands of other neurons, and you got billions and billions of them, it's just an unfathomable amount of connections that you can make inside those neural networks. So the nervous system transmits. So they're connected by the axon. Yep. The axon is the key component. That's the electrical cord. That's the place that sends the message. So yeah, so the nerves are all axons. Yep. So in the nervous system, we're sending neurotransmitters. In the endocrine system, we're sending hormones. That's really the only difference between the two. I put that on I think I put that on your easy to mix up terms. The difference between the nervous and the endocrine, and really the only difference is the neurotransmitters versus the hormones. They both transmit messages. They both transmit messages across the body. Just the nerves do it through the axons of the neurons, and the endocrine system does it through the blood with hormones. Nerves, these are just cables, just lots of axons, part of the peripheral nervous system. We've got the somatic nervous system and the autonomic nervous system. These were two nervous systems that were explained in the chart that I handed out in class today. So the somatic is the division of the peripheral nervous system that controls the body's skeletal muscles, and the autonomic is the division that controls our glands and internal organs. So autonomic, obviously pretty easy to understand. You don't have to tell your heart to beat. You don't have to tell your lungs to inflate. You don't have to tell your stomach to digest. That's all automatic. So that's part of the autonomic nervous system. You do, however, have to tell your finger to move. You have to tell your mouth to move, like I'm doing right now. You have to, so the muscles of your body's skeleton are part of the somatic nervous system, and that's not automatic. And then sympathetic and parasympathetic, we've covered this in several different units again. Sympathetic is arousal, parasympathetic is calming down. Think of parasympathetic as the parachute that kind of opens up and allows, kind of calm and allows you to land safely. But when you jump out of the plane, everything's hectic. You're skydiving, you're in your sympathetic nervous system, and all of a sudden you activate your parasympathetic, the parachute opens, and then it's a nice, calm, gentle return down to the earth. So I'm going to go back here. And these were in the chart as well, right? Yeah, this is what was outlined in the chart. So these are all part of the peripheral nervous system. So you got this chart again in class. So I'm just kind of explaining that a little bit in more detail. So that's sympathetic and parasympathetic. So the sympathetic arouses, and that's all the stuff on the left. Again, you got this chart again, because I think this does a really good job showing all the different autonomic functions that the sympathetic and parasympathetic control. So take a good look at that, and if you have a good understanding of this, then I think you have a good understanding of sympathetic and parasympathetic. This is just another diagram that shows that. So central nervous system is just the spinal cord and the reflexes. So your sensory nervous system is part of your peripheral nervous system, sends incoming information into your central nervous system, which is just part of your spinal cord. And then that takes it up to your brain. So that's just a quick overview of the nervous system itself, all the different types of the nervous system, again, outlined in that chart that I handed out in class today. So a major part of this unit is how do we know what we know about the brain? And this is an area that I think people struggled with on the exam. And really, again, there's not much. When we talked about Phineas Gage, we talked about clinical observation and doing autopsies on people who had problems and looking at their brain. We talked about ethics, whether it's okay to create lesions in animals' brains just so we can study the results. Is that okay? Obviously, we know we can't do that on humans. We talked about Phineas Gage, why he was so important, because he was a traumatic brain, really the first documented traumatic brain injury case where he actually had a lesion in his brain. So really important. So unfortunately, we can't go in and open up people's brains to observe them. So we have to figure out a way to do that from the outside. And there's 4 main ways that we do that. And it's these 4 things that you do need to commit to memory. These 4 things are probably going to appear on the exam, and you may even see one of them in an FRQ. So the first one is the electroencephalogram. All that does is it shows the brain's activity. It shows a functional reading of the neural transmissions that are happening in the brain. So it senses the ions that are passing in and out of the axons and creating the action potential. It senses those, and it feels it can outwardly graph how much activity is occurring in which parts of the brain. That's all it does, is it just reads the electrical waves that are happening on the brain surface. So that's it. Electroencephalogram. It's really a non-invasive way to get a good reading of what's going on inside the brain. But unfortunately, you need to be a trained reader of EEGs to be able to figure them out, because this is what the reading looks like. So you really need to know what you're doing in order to figure out whether things are normal or not. Then we've got MRIs. These are a little bit more invasive because you have to actually put your brain into a machine that then reads it, and it reads it by sending these magnetic fields or radio waves basically into your brain, bounces them off of your brain. And the only thing that this shows is it shows tissues. So it shows the brain tissue. It can show whether there's a lesion in your brain. It can show whether there's a dead area of the brain. It's used on stroke patients to figure out what areas of the brain have been affected by strokes. It's used on Parkinson's patients to figure out if these neurons are actually pruning themselves. You can see dark spots inside the cortex to figure out what's going on. But that's all it shows. It just shows tissues. Then we've got PET scans, which show brain activity. They don't show tissues at all. So if there's a dead spot in the brain, the only way you'd be able to tell on a PET scan is that area wouldn't light up as well. But it doesn't necessarily mean that it's dead. So a PET scan just reads the radioactive glucose that gets injected into your bloodstream. And we know since neurons are cells, they need glucose for energy. And so they absorb glucose. So the PET scan and the MRI that gave us the first real images of just the mic is cutting out. It keeps muting. Bummer. Shoot, I'm gonna switch back over to my old mic. Okay. Is this good? It's not. Oh, okay, it is showing. All right, so I switched back over my old mic. It says that I'm transmitting. Can just one person let me know if you can hear me? Very good. Yeah, okay, good. And I guess Emily, if you've got something I can borrow for future sessions, that would be awesome, because my mics here don't seem to be of very good quality. So sorry about that. Awesome, thank you, Emily. So yeah, so the CT scans. Hopefully you got enough information that it didn't cut out enough, that you got the information about the PET scan. And the CT scan does a little bit of both. It does tissue and brain activity, but it doesn't show as much brain activity as the PET scan does. And the key with the CT scan is that the X-ray rotates around the head, so you get this awesome 3D image. That's the one I showed you with, like, you can look at the different layers and you can see really good details of the bone and soft tissue that exist. So those are the three that you need to really, or the four, I'm sorry, four that you really need to focus on. The EEG, the MRI. The EEG just shows electrical impulses. The MRI just shows soft tissue. The PET scan shows brain activity, and then the CT scan shows bone and soft tissue, but it does it in a 3D manner. And you can get a combination CT PET scan. You know, so there's different combinations of all of these, but those are the basics. So those are the four that you need to know. Good? Good. All right. Now we're going to talk about the brain. Some brains are bigger than others, as we see Homer here. very tiny brain, which you can get from his personality. The AP, I was looking through old AP exams in preparation for this, and I saw that they referred to the brainstem as the old brain, and they refer to the cortex as the new brain. So when you see something that refers to the old brain, know that they're talking about the brainstem. Why do they call it the old brain? It's because it's the oldest part of the brain. In fact, right when the embryo starts to divide, the first few cells that start to divide are brainstem cells. So it's really the oldest part of your body. So it's oldest, meaning that that it's the first part of your body that started to develop in the embryonic stage, really the zygote stage. So when they talk about old brain, you really kind of keep that in the back of your mind. Old brain, they're talking about the brainstem. Remember, without any of these things in the brainstem, you can't survive. So you can have brain injury all over your cortex region. You can even have some brain injury in your limbic system, but you cannot have any brain injury inside the brainstem. If you do, you will die. These are very, very vital areas of the brain. You can have some damage in them. There are some stroke patients who have some damage in these, in a little bit of these areas, but if you lose any one of them, then that's it. You're done. So the brainstem is as important to the functioning of the body as the heart and the lungs. Can't live without it anymore. So these are the areas that there are. There's the thalamus, the midbrain, the pons, the medulla, spinal cord. The pons. Now, how much of this do you have to know? Well, this is where you kind of have to make some judgment calls here. Do you really want to spend all of your time going over your brain anatomy and learning each individual part of the brain, or do you just want to, in my opinion, if I was studying for the AP exam, I would just kind of review these things, just try to keep them as fresh in the memory as possible. Maybe make some index cards that you can look at right before you go in to take the test. But I wouldn't be sitting there making charts and committing it to memory and coming up with different tricks to try to remember all these different different parts of the brain anatomy, because the whole brain anatomy section, I think it is the whole neurological section, neuroscience section, is 8 to 10% of the exam, and then if you take the brain anatomy section of that, it's probably maybe a third of that. So you're looking at maybe 3% of the exam that could possibly have questions on brain anatomy. So I don't know if it's, and it would take a lot of studying to commit all of these to memory. So I think just like a review tonight, like I'm going to do, move quickly through them, and then the review that you're going to have as you look this stuff over before the exam, it is good enough. I think it would be a waste of time to sit here and actually try to remember all of this stuff, because it's difficult. So don't feel stressed out if you're going into the exam and you're not quite sure what the pons is. Don't worry about it. You may miss a question, but again, you don't need to get them all right to get a 5 on the AP exam. So I wouldn't freak out sitting down at the test and saying, "Oh, what was the reticular formation again? Shoot, I'm going to fail the test." No, not going to happen. So take a deep breath on these brain anatomical structures. So the pons does. It coordinates movement, and it integrates movement on each side of the body. So the pons is, so really any movement that you do has to go through the pons. Reticular formation is really important because it keeps you awake. If there's an abnormality in reticular formation, it causes narcolepsy. We looked at that cute little narcoleptic dog that kept falling asleep throughout the day. So that was a malfunction of the reticular formation. So it controls arousal. Then the medulla is probably the most important part of the entire brain that controls the heartbeat and the breathing. Without the medulla, you're kind of in big trouble. The cerebellum is that area behind the brain. It almost looks like a clamshell. There's one on each side, and that's called the little brain, or it's nicknamed the little brain, and that helps coordinate voluntary movement. So your motor neurons go down through the cerebellum, and it makes it so that your voluntary movement is fluid and kind of in tune with what with each other. All your muscles are working in tune with each other. So it's a really large area of the brain, and it's shaped and structures is a little bit different than any other part of the brain in that little clamshell structure that exists right behind the brainstem. The limbic system is like this donut-shaped system that sits on top of the brainstem, and that contains a lot of the areas that we've talked about, a lot of the different things that we've talked about that have functions in other areas of psychology. So we talked about the drives such as fear and aggression that happen in the amygdala, and the drives for food and sex and those kind of things all come from this amygdala. Fear, anger, the amygdala, which sits in the limbic system. The amygdala is just a little lima bean, one on each side, and that covers fear, anger, and disgust. The thalamus we talked about being the switchboard of the brain, and any sensory or motor information that enters or leaves the brain goes through the thalamus. So really, really important. The hypothalamus sits below the brain. That maintains maintenance activity, or it directs maintenance activities like eating, drinking, body temperature, control of emotions, those kind of things. So the hypothalamus is extremely important. Hypothalamus just means that it's sitting below the thalamus. In the hippocampus, this is extremely important in the creation of new memories. All memories are kind of, it's basically the switchboard of memories. It tells the memories where they're going to put them. It tells the brain where the memories are going to be stored, and then when you go to recall the memories, you're actually activating the hippocampus, and the hippocampus is going out and grabbing that information from other areas of the brain where it's stored and retrieving it in for your recall. Also, this is the only place that new neurons develop. And then the cerebral cortex is everything else, everything outside of either the brainstem or the limbic system. It's divided into 4 lobes: frontal, parietal, temporal, and occipital. The only thing really in the occipital lobe is your visual cortex. That's the only thing that really, well, the entire cortex is responsible for thinking and storing memories. But the only structure that, the only specific use of the occipital lobe besides that is the visual cortex. The other 3 lobes kind of interact with each other, and their uses are kind of blended in together. In the frontal lobe, the dividing line between the frontal and the parietal lobe is the dividing line between the motor cortex and the sensory cortex, which lays across the top of the brain there. We know the motor cortex, responsible for carrying messages out to your muscles. The sensory cortex is responsible for receiving those messages back from the body. And they sit right next to each other on the top of the brain. And we talked a lot about the prefrontal cortex. Hopefully you have a good understanding of what the prefrontal cortex does. It's involved in planning, complex cognitive behaviors, personality expression, decision-making, moderating correct social behavior, all those things. And it was the prefrontal cortex that got severed in Phineas Gage, changed his personality, and made it so that he didn't have correct social behavior, those kind of things. And we talked about doing a frontal lobotomy today in class. When you take the frontal lobotomy out, you take out personality expression. So you're removing that personality disorder that somebody had that was untreatable with disease or with medication. And we've got Broca's area and Wernicke's area. Broca's area is located actually in the frontal lobe. A lot of people get that mixed up. They think that both of them are in the temporal lobe, but only Wernicke's is in the temporal lobe. Broca's is actually in the frontal lobe, and then the occipital lobe is just the visual cortex. Really cool MRI scan down, a functional MRI scan down on the bottom, which is the functional MRI is the PET scan combined with the MRI. And that shows the occipital cortex, the visual cortex in the occipital lobe lighting up when somebody sees stuff. Parietal lobe, see, I don't know how to do it. I know I'm going really fast, but again, I just don't know. I'm overwhelming you with information right now, but hopefully some of it's going to sink in. I feel bad doing this, but we just got to get through the brain anatomy. Again, I'm not sure if this is the best way to do this, but if anybody has any suggestions, let me know. So, so the sensory cortex, again, is the dividing line between the frontal lobe and the parietal lobes. So that is where the sensory information comes in from the body. And this is another look at that image where we know exactly what parts of the sensory cortex are responsible for what sensations, which is really kind of cool. We can do that through PET scans. We can figure out which areas of the sensory cortex light up when, say, your hand gets touched or your eye gets touched or something like that. And then in the temporal lobes, we've got this Wernicke's area, auditory cortex mixed with Wernicke's area, sitting right basically above your ears. So the auditory impulses go through your ear, up into the information processing area, and then back up into the auditory cortex. And it also contains Wernicke's area. We spent a little time looking at people who suffered with suffering Broca's aphasia versus Wernicke's aphasia. And remember, Broca is responsible for understanding, and then Wernicke is responsible for expressing. So those are two typically mixed up terms, Broca's and Wernicke's. Just remember, Broca's deals with the information coming in, Wernicke's deals with the information going out. And then the hemispheres. We looked at a great TED Talk by Jill Bolte Taylor, who was the neuroscientist who had a stroke, and it was attacking the right side of her brain and interfering with the processing that happened on the right side of her brain. But she was a scientist, so she was a right brain person. So she had to switch her whole life. She had to give up her career, and now she does art because she's a left brain person now. So she switched from the right to the left. Fascinating TED Talk about a neuroscientist examining her brain as it's undergoing a stroke. There's no better way to describe the hemispheres and how they work than through the life of Jill Bolte Taylor. So the hemispheres, we say that they're contralateral controlled, which is the left brain controls the right side of the body, and the right side of the left side of the body is controlled by the right side of the brain. We say right side dominant people are better at spatial and creative tasks, and the left side are better at logic. Did I just screw that up? I did just screw that up. I said yeah, I'm sorry. I said that Jill Bolte Taylor had, she was a left side person. She was a logical person. She was a scientific person. And the left side of the brain is what's required if you're a logical, mathematical person. The right side of the brain is the creative side. So she switched from the left side to the right side. No. Yeah. Yes, we're good. So that was the way we did that. Yes. Okay. So we talked about right brain functions being the creative side, the intuition, the insight, and then the left being logical, mathematical, language, science, math, those kind of things. And of course, if you're Homer Simpson, this is what your brain looks like. We looked at that girl who had those epileptic seizures, and the seizures were caused by the random neural firings that happen across both hemispheres, and happen through this part of the brain called the corpus callosum, which is the bundle of axon fibers that carry connections between the two hemispheres. And the only way that they could fix the problem was to actually sever the corpus callosum, so the two sides of the hemisphere, the two sides of the brain didn't communicate back and forth. What's really interesting is if you have your corpus callosum severed, whatever you see from your right eye is interpreted by your left hemisphere. Whatever you see by your left eye is interpreted by your right hemisphere. So the different functions of the different hemispheres are involved in the different objects that you see. I think I've got a image of that coming up. Severing of the corpus callosum, talked about that. So we found there's no loss in intelligence if somebody severs the corpus callosum. Most people can no longer remember dreams. They have trouble putting faces with names. Spatial notation, depth perception are off, but that's really all that's it. So yeah, so here's the image here. So your left visual field is interpreted by your right side of your brain, and then your speech side of your brain is on the left side. So anything that comes out of the right side can be spoken. But anything that you're seeing with the left side of your brain, you can't say what you're seeing because it can't communicate with the speech areas of your brain. Anyway, we know that hemispheric specialization, talked about that, hemispheric dominance. Again, just a repeat. This is an object that's supposed to tell whether you're left side or right, left brain or right brain, which face is happy, which one is sad. Whichever face is happy is supposed to be your brain dominant side. So if you're seeing the left one as happy, that means you're a right brain dominated person. If you're seeing the right one as happy, then you're a left brain dominated person. But most people just see them both as the same. I don't know. The last part of this unit was the endocrine system, and that's how the body communicates through blood and hormones. So we've got all of these parts of the body that are involved in the endocrine system, all have specific jobs. So what are hormones? They're just chemicals synthesized by the endocrine glands. They secrete into the bloodstream, and they direct parts of the body. They direct all the different organs and tell them what to do. For example, epinephrine increases heart rate, blood pressure, blood sugar, feelings of excitement during emergency situations. It's kind of like opening a floodgate. You just put the hormone out into the blood, and then it automatically influences everything that the blood touches. So only certain cells in the body can respond to hormones, and only at limited times. So in order for cells to respond, they must have hormone receptor molecule in order for them to pull the hormones in. An example of that would be the hormone of oxytocin. It's only released during a specific, well, it's released all throughout a person's life, but it's only affected when a woman has just given birth. Causes the uterine contractions to end pregnancy and causes breast tissue to eject milk. So that's a really good example of a hormone that is in your body at all times, if you're a woman, in your body at all times, but only has specific jobs at specific points in your life, only right at the end of pregnancy. The pituitary gland is a master gland. That's the one that controls everything. So you can think about the hypothalamus being the switchboard of the brain. I'm sorry, the thalamus being the switchboard of the brain. The pituitary gland is kind of like the switchboard of the endocrine system. So obviously you would think that the thalamus and the pituitary gland have to communicate with each other, which they do. They communicate through the hypothalamus. So the thalamus is the switchboard of the brain, communicates with the hypothalamus, tells the pituitary gland what to do. Then the pituitary gland comes out and tells all the other glandular organs of the body what to do. Thyroid, parathyroid, this involves metabolism. We talked about diet and bone health, not something I think you need to know. Adrenal glands. You know what? I'm going to skip over. Oh, shoot. I thought I had a diagram of all the different hormones. Did I give that to you today? See if that was that in the review sheet I gave you today? It wasn't. Okay. So just like with the neurotransmitters, I think I have a slide of all the hormones that you need to know. I don't understand why I didn't make it into this slide presentation. So sorry about that. Let me go back. What's the pituitary gland regulate? It regulates all the other glands that secrete hormones in the endocrine system. So the pituitary gland is like the brain of the endocrine system, and it tells all the other organs what to do. So when you think, if there's something going on with your endocrine system, if there's hormones being secreted, chances are those hormones, they may not be directly secreted by the pituitary gland. Whatever gland secreted them was probably told by the pituitary gland to do so, if that makes sense. So really nothing happens in the endocrine system without the pituitary gland first telling another gland what to do. But the pituitary gland does communicate directly with organs such as the kidneys and the muscles, and the breast and the ovaries. The testes are a gland themselves. They secrete the sex gland, sex hormones. And then the thyroid secretes the hunger hormones. So, as far as the endocrine system goes, there are some hormones that you need to know. There are some, I mean, you have to know the glands. This is a tough one. I'm not really sure how to direct you on studying because any of this information that I just presented to you could pop up at any time on the AP exam. So you have to study enough of it so that you're comfortable and familiar with it, with the different glands. I don't know that I think I would just try to link the glands with the hormones. And then I would study the hormones. So kind of break it down that way. So figure out what the hormones do. And those I think you have to commit to memory. I thought I had a chart. I'll try to get one together for class tomorrow, or class on Friday. As far as what the hormones do, those are just like with the neurotransmitters, those are just some things you have to commit to memory. And then just know what glands are responsible for which hormones, and then I think you're good to go as far as the endocrine system goes. But again, we're talking about a small percentage of the exam. So if you don't have time if you don't have a large chunk of time to dedicate to the endocrine system, I don't know that it's going to harm you a ton. So yeah, so I'm sorry I can't help you more on the endocrine system, but it's just a tough thing. There's just some things you have to memorize, however you see that fit. All right, so I did it. We did it before 9 o'clock. There is a section on development in this unit. I don't know that anybody wants me, I don't know that I want to continue talking about development. But you do have to know the work of Piaget, Erikson, and Kohlberg for the Ainsworth parenting styles. So the key psychologists for the developmental unit are Piaget's schemas and Piaget's stages. Ainsworth's parenting style, we talked about authoritarian, authoritative, and permissive. Harry Harlow's monkey studies, where the monkeys either were attracted to the mother. They went to the wire mother for food, but they went to the cloth mother for comfort, and they stuck with the cloth mother. So we learned that children don't connect with their parents just because their parents provide them food. They connect with them for comfort. So we've got Harry Harlow, and then we've got Erikson's stages of development, and then Kohlberg's stages of morality. So those are really, I think there's 5 people I just listed that you have to know for the developmental unit. I could go over them now, but I don't know how effective that would be since you've just listened to me for an hour on some pretty weighty stuff. So I guess if there's some gung-ho people who are willing to tough it out, I could quickly go over Piaget, Ainsworth, Harlow, Erikson, and Kohlberg. I'm ready to do that, but I'm not offended at all if you log off and say, no way, Mr. Jarvis, I'm overwhelmed. My brain hurts. So maybe I should take a poll. Oh, I can take meeting notes? I didn't know that. All right. This is going to dictate what we do. Here's the poll. Wow. All right. So 83% said, let's do this. All right. Cool. So I'm all for it. 2 people said, good night. And again, I'm not offended. You've been listening to me for an hour. For those 2 people who said, and I don't have names. Names aren't associated with the poll, so I don't know who they are. 2 people said they're going to bug out. Again, I'm not going to be offended. You've got your credit. You went through the most difficult part of the unit, so good night. Go ahead and log off. For those of you that want to tough it out, let's do it real quick. All right, developmental psych. In the beginning of developmental psych, we talked about prenatals and newborns. Asked you these great questions. What do you like about being your age? What's the worst thing about being your age? How old is too old? Those are the types of questions that developmental psychologists deal with. These are the major issues that are covered in developmental psychology. We've got nature versus nurture. Obviously, that's a question that we talk about, or that we try to answer at every single unit. Is what we're looking at a result of nature or nurture? The issue of continuity versus stages. So is development gradual, a continual process, or a sequence of separate stages? Like Piaget says, once you get to a certain point, your development jumps up and you immediately enter this other stage. Whereas Erickson would say that's not true. It's kind of real. It's a real gradual, and you can't really tell which stage someone is in because they can be partly in between one stage and another. Piaget would say that's not possible. You're either in one stage or you're in another, and your development jumps up in these sequences of separate stages. And then the key here is a question that we try to struggle with is your developmental psychology. Is it stable? Does it persist through life, or do we become different persons as we age? Those kind of things. So we spent a long time talking about conception, and we looked at the conception video, and there's this idea of the prenatal development, the newborn. All you have to get from this section right here are the key vocab terms. What is a zygote? A zygote is just a fertilized egg up to about 14 days old. At 14 days old, it turns into an embryo. And then at 9 weeks, the embryo turns into a fetus. So just zygote, embryo, fetus, all stuff that you learned, I think, in wellness. And then there's teratogens that can enter into the developing baby. One of the worst teratogens that could possibly be entered through the mother's blood is alcohol. And so an alcoholic definitely will have a child that's born with some major problems, because alcohol is a nasty teratogen. HIV is also another one. Most or some mothers who have HIV can have children that have HIV, and then any other kind of drug use. What's really cool is that when children are born, they're born with some sort of reflexes, like they know how to eat, they know how to locate food, they know how to suck, those kind of things, which is kind of cool. So that's definitely nature. So we know how to get mother's attention. We know how to cry when we're hungry, those kind of things. That's nature. Maturation is just the process of you going through these subsequent stages in order to get to be who you are. We talked about these developmental landmarks. I had you guess to see what the landmarks were. Not really important. These are just some developmental landmarks. This just shows maturation, that things happen in steps and stages. And we go from that to child cognitive development. This is where Piaget is. In Piaget stages. So who was Piaget? He was a guy that was in charge of norming psychological tests. We talked yesterday about the norming and the standard bell curve. He was the one that was in charge of making that center line, figuring out where that center line is on some psychological tests. But when he was doing this with children, when he was doing these tests, he was thinking he became intrigued about how similar the wrong answers were, that children weren't just making up wrong answers. There was a systematic procedure that all of these wrong answers were very similar to each other, and their thinking was the same. So he came up with this idea that children don't just know less, they know differently. So that led him to create his idea of schemas. And then we went into these ideas of simulation versus accommodation. So simulation is the interpretation of new experiences into present schemes. That's where my daughter thought the animals at all the animals at the zoo were puppies, because the puppy was the only four-legged animal she'd been exposed to. Versus accommodation. Accommodation, when she got a little older, she realized that the zebra is not a puppy, and she made new schemas, she modified her present schemas to fit those new experiences. So simulation is trying to fit the new experiences into what you've already got. Accommodation is actually changing what you've got to fit the new information. This is just my mom with a DVD player, daughter to the zoo. Now here's Piaget's stages. These are, if you are going to spend a little time, the development unit isn't packed with very many of these, and the development unit I think is a 7 to 9% unit. So it's a pretty decent chunk of the AP exam. So these I would spend a little bit of time studying. There's not that many of them, but Piaget's stages, we've got birth to 2 years at the sensory motor stage and object permanence. Really, that's, if you know sensory motor stage, birth to 2 years, object permanence. Those are the kind of things that you have to link together to get Piaget's first stage. Stranger anxiety also happens in the sensory motor stage. And then you've got pre-operational, 2 to 7 years, and you've got conceptualization. So it's really when you've got, they start to use symbols, they lack the concept of conservation. Oh, that conservation, when we looked at the videos of the kids who had the equal amounts of water in 2 different vessels, and then you pour the water into a taller vessel and they automatically think that that vessel has more because it's taller, or you spread out the coins and they think the longer line has more coins even though they have the same amount. So it's that law of conservation. And egocentrism happens in this stage. So so the pre-operational stage, they're just basically lacking conceptualization. They're having trouble conceptualizing and perceiving different higher level thinking things. So these are the types of tricks that you can play on a child who's in this stage. The egocentrism, you ask a child, why does the sun shine? Why is the grass green? The sun shines to keep me warm, and the grass is green because it feels good on my feet. That's the whole reason that there's green grass in this world, is because it feels good on my feet. The sun is shining over this entire world because it feels good on my face. Daddy must eat goldfish and chicken nuggets for lunch because that's what I eat. That's what I would want to eat. Then we move into concrete operational. This is where you can focus on more than one aspect of a problem at one time. You're able to debate both sides of an issue, and accommodation becomes easier, happens at a faster rate. So you start building these more complex schemas in your mind. And so you start to get conservation of number, conservation of mass, those kind of things. And then the functional operational stage, which is what all of you are in right now, that's where you can finally think abstractly, hypothetically. You can experiment with different outcomes, use analysis, reflection, analogies, those kind of things. So that's really the final stage of Piaget. So what are the essentials? Children develop reasoning abilities through active involvement with their environment. They fuse new information into existing knowledge, and specific reasoning abilities and limitations characterize different age levels. And Piaget is the one that says you're moving along basically at a flat line, and all of a sudden you hit this point in your development where you jump up and you move on to the next stage automatically. All of your thinking and reasoning abilities is onto that next stage all at once. And that's kind of a criticism of Piaget. That, but again, Piaget is a lot like Freud. Piaget did for developmental psychology what Freud did for therapeutic psychology. He laid that groundwork. And although we don't practice a lot of Piaget psychology, we're very thankful to what he did, laying the groundwork for opening the door to developmental psych.
So now we believe that developmental psych is this continuous process, that it's this gradual line, it's not these jumping up of stages, and that children express their mental abilities and operations at an earlier age than what Piaget said, and that formal logic is a smaller part of cognition than what Piaget made it seem. Piaget said that basically formal logic is the end-all be-all. Once you get to that point, you're a solid human being, but we know it's a little bit less than that. Would you need to know the criticisms of different psychologists? I think you would, just because it helps you understand their theory a lot better. I think once you understand the criticisms, you can really wrap your entire head around. You can wrap your head around the entire theory that they had. If you just understand their theory, I think you don't have as deep of an understanding of what it's all about. Are you getting a specific question on a criticism of Piaget? I doubt it. I'm not going to say with certainty, though, that you wouldn't get a question on this. This is fair game. This is definitely in the AP Psych curriculum. I don't remember ever seeing a question like this, but that doesn't mean that it doesn't exist. So you could possibly get a question on the criticisms of different psychologists. But I think knowing this information definitely, I wouldn't sit down and memorize each one of these to know. I wouldn't quiz each other. What are the criticisms of Piaget? Number one, but just going over it and knowing it, including it with your reading or your review, definitely will help you get a better understanding of the philosophy itself. All right, so attachment. This is the Ainsworth study. The Harlow study. Sorry, Harlow. Ainsworth is with parenting. So it was first assumed that infants became attached to those who satisfy their needs. So that's why infants are so attached to their moms, because their moms give them the nourishment. But Harlow was the one that kind of refuted all of that. He said that, this is parenting, I don't know why that's in there. So with his study of monkeys, I thought we had more information on Harlow's. Oh, I guess not. So there's not a whole lot on Harlow here, but he's the guy that we watched the video. I think that's why, because I took the video out of this. But we watched the video where we had that wire monkey and the cloth monkey, and it's that idea that it's not the one who gives you food, it's the one who gives you comfort that you bond with the most. So it's that idea of attachment, which adults do children attach to? The ones that give them the most comfort. So because of that, we look at unresponsive and responsive parenting. The unresponsive parent is the wire monkey that just gives the child food. It's inconsistent parenting, little time for the children, and we know that there's some effects that can happen. The people who are raised by unresponsive parents tend to be opportunistic in their reproductive strategy. They emphasize quantity in mating. They have early sexual maturation. And then those who are given responsive, sensitive, consistent parenting, these are the ones who are raised by the cloth monkey. They get secure attachment, and their reproductive strategy emphasizes quality. There's late sexual maturation, and so we know that there's long-term effects of attachment. And we talked a little bit about imprinting. Can imprinting happen with humans? Imprinting is just where one species imprints on another. It's just whoever you connect with during that critical period of development. And so this is just a cartoon of the psychologist who actually gets imprinted on the ducks rather than the ducks getting imprinted on the psychologist. And we looked at the feral children and the children who were raised by wolves, and they actually imprinted on wolves. And there was that girl in Czechoslovakia who was barking like a dog and stuff. So imprinting, we know, can actually happen with adults or with humans. Just switch in here to there it is. So parenting, we talked about attachment with parenting, and this is the Ainsworth study. So she studied children. She'd place a mother and a young child in a room, then she'd ask the mother to leave. And then through naturalistic observation, they would study what happens with the child when the mother leaves and what happens when the mother comes back. So, oh shoot, this one didn't, this one didn't convert properly. But it was found that 66% of the kids had secure attachment, where they would go to the mother for comfort. They would cry when the mother left, happy when the mother returned. Then we had insecure avoidant, not distressed at the mother leaving at all. There's a cool response when the mother returns. This is probably caused by very distant mothers. And then there's insecure resistant, which is very clingy. They're traumatized by every stage of the process. And when the mother returns, the mother has a really difficult time calming the child down. So the mother doesn't bring the child a lot of comfort, but yet when the mother's not present, the child gets upset. That only happens in about 12% of the cases. That's the most dangerous long-term to someone's development. Most children, though, 66% in the Ainsworth study were determined to have this secure attachment, which is good and healthy. This is where we talk about Harlow's monkeys. So they either cowered in fright or acted extremely aggressive. They couldn't mate. These are children who had monkeys who are deprived of attachment. We looked at studies in daycare. Daycare has no long-term effect. Then we covered the three styles, the three parenting styles: authoritarian, permissive, and authoritative. And Homer is the permissive parent, allows kids to kind of experiment and come up with their own conclusions. It's shown that these children have less respect for authority. They get into trouble more often. There's authoritarian. This is the drill sergeant parent. These kids have low levels of creativity and problem-solving skills. They're very passive. And then there's the authoritative parent, which is Marge, open to discussion, exert control by setting rules, but explaining why those rules are created, those kind of things. And it's shown that these kids are well-adjusted, better problem solvers, have good long-term romantic relationships and stuff. So that's the type of parenting style that we all want to try to emulate. And we get into Erickson real quick. We talk about adolescence, adolescent maturity, brain development. Not something I think you really need. Oh, these are two terms that you probably will need to know. Primary and secondary sexual characteristics. The primary sexual characteristics are the parts of the body that are directly involved in reproduction, whereas the secondary characteristics are really what we determine the difference between a boy and a girl is, kind of the outward expression of that. But they're not directly involved in reproduction. So it's what you could use to tell the difference between a boy and a girl, but things that are not directly involved in reproduction. We know the frontal cortex doesn't develop very quickly in adolescence. That's why their thinking and their behaviors are guided more by their amygdala, not the long-term planning of their frontal cortex. So you can't really blame a 17-year-old kid who makes a stupid decision because it's the prefrontal cortex's fault, not his. And cognitive development, developing reason, just another Piaget thing. And then morality, this is the key part here. Lawrence Kohlberg talked about the stages of morality, and then these are the stages. Now, he went over these 6 stages, but I think really all you need to know, I wouldn't spend a whole lot of time studying these 6 stages. I think what I would do is study the 3 sections here. We've got the the 3 levels: the pre-conventional level, the conventional level, and the post-conventional level. And this slide right here is the key for Kohlberg. The pre-conventional morality happens before age 9. Children show morality to avoid punishment. Then there's the conventional morality, where social rules and laws are upheld for their own sake, just because a rule is a rule, and that's what we need to do. We need to follow the rule. And then we finally, hopefully, eventually get, according to Kohlberg, to post-conventional morality, where this affirms people's agreed-upon rights or follows personally perceived ethical principles where you're doing things because it's right, and it may mean breaking the rules. Doing what's right is not always following the rules. Sometimes you have to break the rules to do what's right. Just for example, Rosa Parks, she got arrested, she did what was wrong, but she was doing it because it was right. So that's that post-conventional morality, that stage that everybody hopes to get to at some point. And they can be broken down further by different stages, the 6 stages here, but again, I wouldn't spend a whole lot of time studying that. I think if you know that Kohlberg deals with morality and their stages, and maybe what the different stages deal with, you should be good. Just a kind of a preliminary study of Kohlberg. That all humans face difficult moral decisions. All humans handle these decisions differently. We progress from stage to stage. What's interesting about Kohlberg is he's the only psychologist that said that you can go back and forth. You can progress and regress through your stages of morality. So once you get to stage 3, it doesn't mean you're going to be stuck there. You could go back to stage 2 for your next moral decision, and then you can jump up to stage 5 for the next moral decision. What he would like to see is that you progress slowly up in stages throughout your entire development, but just because you regress and go back to an earlier previous stage doesn't mean that you're not a moral person. So that's Kohlberg. There's 2 last, but this one's really quick, I think. This one is the idea of forming an identity. Yeah, this is real. There's only 6 slides. Forming an identity is what adolescence is all about. That's what happens in adolescence, trying to figure out who you are. And there's a lot of different influences that happen, that are a lot of different pressures that are put on a teen as they're trying to figure out who they are. It's the first time they become independent of their parents. They have to choose their own religiosity, their own career. They have to do this all the while maintaining peer approval, relationships, really tough time. This is where Erickson steps in, and these are Erickson stages. And I gave you these in the handout that I gave you in class today. So as far as this one goes, yeah, there'll probably be questions on Erickson. How you want to handle those? How do you want to study for those? I don't have a whole lot of advice for you. It would be nice if you could get this whole chart into memory, but is it the best use of your time? I don't know. You're going to have to make decisions like that the whole time you're studying for AP. It's impossible to get all of the AP curriculum into your head and ready to be spewed out on a test. Well, I shouldn't say impossible. I've had a couple of students who I was pretty confident they had all the material in their head, but it's pretty rare that you'll be able to do that. Just get as much as you can. This is a chart you should definitely review. Maybe it's a chart you want to take with you. Yeah, I bet you M would do it. Yeah. A couple of the M's maybe. This is a chart. Maybe you want to keep it in a folder of material that you look over right before you take the test, just so that you can kind of jog your memory on some of these things. Don't know if it's the best use of your time to commit this to memory. Ideally, yes, but that's a decision you have to make. You have that in your handout that I gave you today, so we'll move on and talk really quickly about adulthood. Talked about a lot about old age. We looked at the oldest living people. 10 verified oldest living people. We've got a 116-year-old person in the United States. As of October 2012. This is an outdated one. The one I showed you was more updated when I showed you in class. We talked about physical development. Peak physical performance happens at the age of 20. After that, you're declining. So you guys are about ready to hit your peak. Make the best of it, because after that, it's all downhill. We talked about women at age 50, somewhere around age 50, going through menopause, no longer able to reproduce. And then after age 70, we know that senses diminish. Muscle strength also declines. We know that older people, well, younger people and older people get into way more fatal accidents than the middle-aged people. From about 30-ish, maybe say 32, 33, all the way up to about 59, about 60. Those are the safest drivers. And then after that, you can see 75 and older, they have a really, really high fatal accident rate. So that just is an example of your sensory motor system shutting down. After the age of 80, we know the chemistry in the brain begins to change. The potassium and the calcium ions, they begin to diminish. So once you have less of those, then there's less of an opportunity to fire action potentials. So the action potentials slow, and it becomes more difficult to think, unfortunately. When neurons aren't being used because the action potentials can't be fired, then the brain prunes those neurons, and then you permanently lose that thinking. So no way to stop it. It's just aging. Two types of intelligence, fluid and crystalline. Hopefully you'll remember these. These are pretty easy. Fluid intelligence is the moving intelligence, the ability to reason speedily. This is something that declines with age, but the crystalline intelligence, think of like a crystal cavern, crystal collection. Its crystals are really hard, dense rocks. And those are the accumulated knowledge and skills that happen over time. You don't lose crystalline intelligence. That stays with you. So it's only the fluid intelligence, the ability to reason and think, that you lose. Chart with that. We talked about adult values. And then we talked a little bit about death and dying. I didn't even include that in here because I don't think it's part of the AP Psych curriculum, but the stages of grief that people go through in death and dying. But that's pretty much it. Whew. So that was a whirlwind 32-minute tour through the developmental unit, all the way from the fertilization of the egg through death and dying. So that's what we go through in our life stages. Again, the key people here are going to be Erikson and Piaget, and then the secondary people are going to be Harlow, Ainsworth, and Kohlberg. But Erikson and Piaget are where the most memorization is going to take place. Kohlberg, just an overview. I wouldn't spend a whole lot of time. And then Ainsworth, just know that that she studied attachment. She built off of Harlow's work and studied parenting styles based on attachment, and Harlow laid that foundation work with the attachment in his monkey studies. Other than that, I think this is a pretty decent unit. I think this is something you can wrap your head around pretty well, not like the previous unit that we spent a lot more time on. All right, so those are the two units you'll be quizzed on. 20 multiple choice questions on Friday, so a little bit longer than the one we took today, because it's covering just a few more chapters. So is there anything else you want me to cover? I'll be happy to go back. If you want to say goodnight, you can go ahead and log off. I'll hang out for just a few minutes longer, see if anybody's got any questions, and then we'll call it a night. I think I'm going to stop the recording right now, just so that doesn't get out of control. And I'll post that link available. I'll post that link out via email as soon as I get it, as soon as it processes a little bit. So you can all review this if you want tomorrow night. All right, any questions? Good, you're welcome. Hopefully we'll have a better mic next time. Sorry about the audio issues. Thanks for hanging in there with that.